Thank you. Good morning. It's my pleasure to welcome you a bit into some research we do in soil science. So if such an animal digs out a handful of soil, maybe a liter, how many bacteria does it move? What do you think? In 1 liter soil, how many bacteria? 1,000 billion? 1,000 billion? And how many nematodes? About 100,000 nematodes. Fungal chain length, if you put the fungi all together in 1 liter of soil?
Kilometers?
Exactly, between 4 and 25 kilometers. So it's quite...
that soil is pure bio.
No. I want to say that the interesting finding is that these bacteria live in a density comparable to humans on Earth, and everything is controlled by space. But it's also quite complex to manage, and this is why we lose food for about 2 billion people every year, which is overcome usually by the use of agrochemicals with risks of environmental pollution. In many countries, especially on the African continent, yields cannot be sustained if you add inorganic fertilizers, so apparently soil functions are lost. And then, of course, we all know that there may be a huge within-field variability of yields between 100 and 0%. And for PhenoRob, it is one aim to forecast these yield patterns and to have an early warning tool to react. So I will put today the focus a bit on understanding the role of soil heterogeneity on plant performance. And this is the way we also structure, in a way, our research we do in the Soil Science Institute, which is the building next to this one. So we have groups working on the environmental fate on priority pollutants. We have a long history in analyzing the fate of different plant protection agents, of antibiotics, of hormones, of other priority pollutants like specific tensides or disinfectants. Since this is not part of the PhenoRob program, I will not tell anything about this today. Main focus is then on the fate of different inorganic and organic nutrients in soil systems. And we have some smaller work to be done on the sensing of these nutrient patterns in arable soils, which I will focus here in this talk today. Okay, when we start understanding the processes controlling the availability or fate of nutrients in soil, we always have the question, at which scale do we place our investigation? In principle, it is the smaller the scale, the more exactly we can describe an interaction. We can define very exactly what is the monobidentate complex, how is a phosphate molecule binding to a mineral phase. But at the end, it may not be relevant to explain yields because maybe they are limited by water, or maybe because water is controlling phosphorus supply, or even other factors. But when we go to the large scale, we have many different processes, and we have to identify first which process is most relevant. And the same applies in a way to the management of the different scales from a soil perspective. What is conventionally done is you control nutrient availability either by direct fertilization or by changing the soil regime, that in a way you alter the bioavailability of the nutrient to the plant. You try to lime the soils in order to have phosphate availability in a nice pH range, or you even add the nutrients directly in the bioavailable form. The bioavailability of a nutrient so that it is available to a plant at the surface doesn't help very much if the nutrient is inside larger structural units. And what is very difficult to assess is the bioaccessibility of the nutrients. Are the nutrients exactly at the place where the plant is growing to, or is the nutrient availability located at the place where the plant is not growing? In addition, we have to concur that the majority of the nutrients is not placed in the topsoil. This is maybe only 30 to 50%. But up to 70% of all nutrients and also of water is below the plow layer. And managing the subsoil is something we try to understand better, particularly when it comes to climate resilient soil management in order to have access to subsoil water reservoirs when the surface soil dries out, for instance. And then at the end, of course, we have the precise management of the fields, which is the main focus of PhenoRob, but all these precise management tools hardly consider these scales in between. So from the nutrient perspective, you mainly look where are nutrients available, where is mineral nitrogen available, but not how it is released from soil structures, etc. So today, what I want to do today is to give you some insights into processes within these different scales. I will start with some studies we do on understanding the nutrient patterns within aggregates, so within three-dimensional structural units. Shortly tell something about work on a profile basis, and then mainly also focus on integrating all these properties when we work at the landscape scale. And at the end, I would like to introduce some work which is mainly done by Stefan Petzold and Gerd Welp, Gerd is also here, on how to use soil sensing in order to get access to this information on the nutrient patterns in the soil. So if there are any questions, I think it's easiest to just interrupt, and then we can discuss it right away. So I will start now with the question, where are the nutrients allocated? When you go from a soil, you pick out an aggregate, and then you destroy the aggregate, maybe by some physical energy, and fractionate it into very little small pieces. I take as an example an average piece range of 100 micrometers, so very small. And the key question we have in our sub-project within this research unit is, if you take out 1 spoon of soil, one teaspoon, very tiny, the same sample, everything is pseudo-replicate, same aggregate, and you take out 30 different pieces of similar size. Do you find 30 times the same, or do we find 30 times something different? And how do we do that? So we isolate these pieces, which is easy. Then we cut them, and then we do an element mapping via electron microprobe analysis, which is an electron beam, which is put onto the surface of the particles, and then they produce an X-ray spectrum. And since it is a quite sophisticated machine they have in the Steinmann Institute, we get a quantitative picture of the element distributions. And when we do that, can be done at different scales, so the very tiny particles down to maybe a size of 10 centimeters. So we have an aggregate, we can embed it into certain metrics. We can use a resin, or we can use water glass. And then you get elemental distributions. This is, for instance, the distribution of carbon. This is the distribution for aluminum. And then we try to use, we call it, well, in a way it's expert knowledge. We also ask people who are specialized in these fields in order to derive the structure from the element patterns. So we know if there's a lot of carbon with wide CN ratio, this is likely particulate organic matter like a root residue. From aluminum silica ratios, we can deduce it's a clay. So in the combination of morphology and element combinations, we can classify these aggregates. So what is inside? Do we have, for instance, microbial carbon spotted everywhere, or do we have different clay minerals? Do we have oxides? And then we get such a chaotic distribution of the different particles. And the question is now, this is defined to a range of 100 micrometers. When we do that not once, but 30 times of the same sample at the same spot, near the same distance of the plant, do we find 30 times the same? And we don't. So everything we do so far in each small aggregate unit, we find something different. Without going very much into detail, there seems to be no rule to understand these element patterns. And at least at the first view, these element patterns are not really reproducible. So what we then did next, we went to Lutz Plümer and asked him, can you derive any information out of these patterns? Entropy information, or whatever information to structure these data. And the first he did was, he did a nearest neighbor analysis just to assess whether at these edges there's always the same compound neighboring each other. And then there's quite some structure in there. For instance, every time we assign a structure to microbial carbon inside these aggregates, we also find plant remains, which makes sense that the microbes grow where the roots are. Then they exude some gums. At the gums, we get negative surface charges, so they can absorb cations. And then you start up building these aggregate units. This is connected to the living space of the microbes. And interestingly, also whether there is lime or not in these aggregates.
Sorry,
yeah.
What does the axis tell me? So to interpret the diagram, what's the x and y axis?
These are, as far as I understand, which is now maybe the people from pattern recognition can help. Now, this is like some kind of principal component, so it's a virtual coordinate. And then you get the distance to the lines, and especially the thickness of the lines tell you something how close they are associated. So if this is a very thick line, they always come together. And also, they may come together via a short distance, but not so often, so the line is not so clear. And the third information we have, for instance, that the presence of silicates is not at all present to the presence of microbial C. Yeah.
Another question, so that means you have no non-automatic method to say what's the state of the truth. So you cannot select that and identify the aggregate without these automatic techniques, that you know what is measured automatically and how to.
Yeah, actually, this is why we are looking also for collaboration. We need people who are able to classify that this goes beyond our expertise. And we don't have it automatized so far.
But if you would do that, I don't know, take this soil and select it, count the number of aggregates, and then chemists would that do in a not automatic way. Would that be possible, or is that not possible? Only to understand it.
Now, we isolate the particles individually, and everything is a non-automated. So the mapping is automatized of the element pattern, but not the preparation of the sample. That's done by hand. Yeah.
Can you describe a bit more how you disaggregate? Because these fracture points would be, let's say, weaker anyway, right?
Yeah.
So something would be driving potentially previous plant roots, the physical weakness at the points. So how do you?
I think in this case we may have. So either we just dry crush it, we just dry sieve it, in order not to disturb in order not to induce any material redistribution in water.
So you let them fall apart?
No, I just press them through a sieve. I force them into a certain size if I want to investigate the size. The classical method is you shake it in water, then let the water disperse these aggregates, and then you may add a different energy of ultrasonic energy, and then you can define at which physical energy, because it's a shock wave, an ultrasonic shock wave, at which shock wave do the particles disintegrate. So we can do that for different stability classes. We can use very high power of ultrasonic energy, we can use very soft power of ultrasonic energy, or we don't use ultrasonic energy at all. But everything we do so far is that the pattern is different on the first view. Yeah.
So this is a two-dimensional picture.
Exactly.
The surface of these microaggregates, or
No, it's a two-dimensional slice.
But it's not the surface, it's somehow.
No, it's the cut. We cut them. We're still working on reconstruction, three-dimensional pictures, because this is a research unit, but anyhow, it looks different, whatever we do. But not the size and not the stability. Well, this is why it's also unpublished. That's why we are working. We try to understand these patterns. We also don't have any clue about their functions. But anyhow, there is some clustering of information that at least within these chaotic pictures, some of these components always occur together. And we think there is some information behind. And now we wanted to understand, does this affect, for instance, the residence time of the carbon in soil or the turnover of nutrients? So what we did then is we built a new machine. This is a laser, which is a laser ablation unit, so that you can look at the specific point within these aggregates. We can shoot through the aggregates, or we can map these aggregates. Then we transfer everything which is shooted away into such a transfer line, and then we can measure the isotopic abundance. And then we can trace the origin of the carbon within these very small scales. And the idea is behind this that if you have a land use change, for instance, from plants with different photosynthetic pathways, the natural abundance of the carbon in the plants changes. So plants discriminate against the heavy carbon during photosynthesis, and they do it at different intensity. So the C4 plants have a final isotopic composition, which is expressed here as a per mil relative to a standard, which is different to plants with a C3 photosynthetic cycle. So whenever we have a land use change from C4 crops to C3 plants or vice versa, we get a change in the input from the plants to the new plants which are grown. And if we know the time of the cropping, we just can use the average composition related to the end members and then determine how much of the carbon has been replaced by the new plant. So this we did now in these aggregates. This is a slide from Klein-Altendorf of the plots where Ralf Pude is working with Miscanthus. Miscanthus is a C4 plant within the C3 soil. We see the signal of the root, which is not really approaching the true isotopic value of the root, likely because also soil particles are on the surface attached of the roots. And then we could look what is the influence of the root on the soil carbon at different distances. And then we see at the distance of about 300 micrometers, we do not detect any carbon from the root anymore. So as an integration, we can conclude the rhizosphere gradient, which affects the carbon turnover in the soil, does not extend beyond 300 micrometers. It's one sample only, but this is the indication you have so far. You can also label the carbon input in the plant and then detect in situ the rhizosphere extension. We also took now aggregates where there are no roots. This is from a site in Seelhausen. It's a site studied in the CRC. It's a bit hard to see, but this spot here, for instance, is the true signal of the C3 crop rotation. Wheat, barley, sugarcane are all C3 plants. And then we grow maize on these plots for 2 years. And then you see there are some spots. Here's the delta value minus 16, which is close to the maize. So 60% of the carbon formerly present in the soil is already coming from the maize. And here 0% is coming from the maize. And we can use this isotopic value to calculate how much of the former C3 carbon, so from the wheat carbon, is being replaced by new carbon from the maize. And then we see there are huge difference within very small distances. And when we convert that to turnover times of carbon in soil, we see that the mean residence of time of carbon varies between 2 years for the spots where 60% is replaced to much more than 100 years at a distance of 100 micrometers.
If you're sampling on this kind of small spots, could also animal carbon or whatever, like old earthworms or nematodes, affect this kind of carbon?
Not very much. So there's an uncertainty of maybe 1 to 2 delta units. But if you have the main signal is the plant. So if you have a field certainly only cropped with C3 plants, also the earthworm signal will have the signal of the C3 plants. It can alter it by 1 delta unit, but that's not very much. So the error is then not very strong. It's a year, maybe, in turnover time estimates. So there are very small hotspots in soil. Carbon sequestration is occurring at very tiny places. I'll come to it. So what we learn from it, we cannot simply disintegrate the soil into small particles and then think we have understood the functions. We have to link the processes in space, chemically and biologically, and this is one task we think should be fulfilled by the professorship in pedogenetic modeling. Not on the small scale, but also then at the large scale and at different time scales. Yeah.
But you showed before showed that the roots only influence in a certain distance, and the question is how many times you need the same species plants to be planted that overall they have this 60% and how many probes you need to estimate that really, because you said that we only have this distance because
Perfect, exactly. The key question is, I think I discussed that with Uli 15 years ago, is a plant growing where there were the roots already? So is the soil in a way the memory of the plant, of the former root architecture, just driving also the next generation of the plant where it grows? Or is the plant trying to avoid, because of allelopathopatic effects or whatever, where the former plant has been? The only thing which we know in terms of how long it takes, it doesn't replace all after 100 years. So we never homogenize the soil completely. But we can take large volumes, and actually if you take a liter of soil, that's gives a good average for this spot. If the spot is representative, because 2 meters apart is again different. Come to that later. With a huge heterogeneity, yeah? And?
One question to your measurements here. You have a really nice lateral resolution, but what is with the time? Is the dynamic? For example, what we know is that you have bursts of exudation of root into the soil.
Yep.
This will directly take down.
Yeah, we're just trying to test their experiments within the priority program where they gas the samples, fumigate them with 13 CO2. And then you see this gradient. It's a similar magnitude after the sampling time. I think that was 2 weeks. But that's the only thing we have. We have only these 2 spots. So it can develop pretty fast. We don't know. I know that Eva wants to extend the method to trace also nitrogen then. Yeah?
Yeah, I was just going to ask about nitrogen. So the turnover studies focus a lot on the ratio of nutrient to carbon in plant versus
Exactly.
microorganisms. And basically microorganisms need more of the nutrient, and that will drive the shift. So can you look at also nutrient ratios?
Yeah, that's what we do.
Spots?
Yeah, we do at stoichiometric ratios. That's how we identified the microbial carbon, based on low CN ratios of 7.
Yeah. So do you see any evidence of nutrient content, I guess, maybe on driving that, first of all, that boundary and the rates, like what I'm just talking about, not at the scale.
We know from other studies, not from this one, that if a soil is nitrogen limited, for instance, that microbes optimize their cycle, release as much carbon dioxide as before in order to get access to these nitrogen sources. So there's a nutrient limitation driven turnover of the organic carbon. That's the only thing we know so far.
Right.
But we do not know the spatial extent.
Right. Okay.
Yeah.
Question. I don't know how much work this microscope isotope mapping is and how you actually do this, but I was just wondering, could you just in a rhizotron put some soil, map all what you see at the window, let some plant grow, and map afterwards and see?
Yeah, that's exactly what's the plan to do. Yeah, you have the rhizotron, then you put little cylinders, and then you can commit. I think the largest cell they had was 3.6 centimeters. The smallest one, it's a sensitivity issue. The larger scale you work, the more volume you have in the total cell of the laser system, and the more air you detect. So it's better to pick out very tiny pieces, but it can be done, yeah. Actually, that was the reason why we built it then. And took us 7 years, then we finished, and then Eva got a professorship and took it to Bayreuth. That's the way it works, but we have permanent access to it. Okay, I would like to come then, what happens at the larger scale? The question also was there. We also know that yield is quite variable. I think this is a biomass estimate from laser scanning done by Georg Barrett within the CRC. And then we can see, for instance, by sampling larger volumes, as said, that we can relate the turnover of the carbon. This is the amount of replacement, how much maize carbon we do attack. This is a figure only for the subsoil. But we see the higher the plants on the surface, the more carbon is also replaced in the subsoil, which makes sense. The turnover of the organic carbon in the arable soil depends on the carbon input. There are many theories to describe it, because microbes, in order to turn over carbon, they need carbon and other nutrients to be able to turn it over. But also the higher is the sequestration. Okay, in order to understand these changes at a longer time scale, we now go away from the very small scale resolution, but we sample the field at different spots, at least 5 times, always 1 liter, then we mix it in order to get a representative soil sample, in order to understand how do soil properties change with time. And what we sample or like to sample in many different ecosystems are so-called false chronosequences, which means you have different sites of different duration of arable land use. So here they started 95 years ago to crop the fields. Then they moved, 60 years ago they cropped another field, etc. And then we have records on the yields within the farmers' books. Then you see that the yields decline in the first 20 years after land conversions, and thereafter they remained stable, most likely because of the introduction of new cultivars. This is not necessarily that the soil is stable. But for us, it's the beauty that you can sample soils of different age of cropping at the same time, the same season of the year, in order to deduce how are the changes in soil properties over time. And we would do that for bulk carbon. For instance, you see the error range is now smaller if you sample representatively. This is a percentage loss of the carbon in the grassland with the time of cropping, that with the loss of yield after 20 years, we also lose a lot of carbon. So we have a rapid loss within 20 years, and thereafter we have a continued slow degradation. Yeah.
Are these from African soils?
This is South Africa, yeah.
And isn't there also slashing and burning going on?
No, this is not in this ecosystem. This is conventional agriculture. These are huge farms, 10,000 hectare size, with 12 to 16 wheels at the tractor. Sometimes they do, yeah, but it's not very often. They're quite afraid of that the fires run away. But actually the burning doesn't really affect the carbon balance so much, because it just burns the grass to ash.
The yield. Before you showed the yield curve. It affects, burning affects the yield.
But only in 1 year, in the very first year. Actually, these slash and burn techniques, they last only for 2 to 3 years if you really do it. So these are really commercially managed fields with fertilizer recommendations, huge input of pesticides. So what we did first, yeah.
A very naive question. Why is carbon in the soil at all important? As according to my naive understanding, plants take the carbon out of the air.
Exactly. And then the plants are decomposed when they lie in the soil, or the roots are decomposed. Microbes grow, and then they form residues. Dead cell walls of the microbes, partly decomposed plants, and that forms the humus, the organic matter content of the soil. But there's not only carbon in there, there's also nitrogen in there, there's organically bound phosphorus, there's organically bound sulfur, at the defined stoichiometric ratio. So if you analyze carbon, you have in a way trace also the behavior of many other nutrients. And then the whole organic matter has certain functions. It can bind the particles together, so it supports aggregation, or you store nutrients, etc.
So the carbon
soil fertility in a way, yeah. It can also be interesting as such for climate change considerations, but yeah. So what we did first is we wanted to understand how much of the carbon has been formed by new crops. These are now rotations between maize and wheat and sunflowers, so we had to add some modeling, and then we see if we do so, even after 80 years, very little of organic matter has been reformed by the crops. So there is no efficient reformation of humus by the new growing crops when you convert the grassland. So we lose soil functions. And we think that this loss of soil functions is related to these aggregates again at different scales, because under the pressure of the heavy machines, they break. Then organic matter inside is released to the air, and with the oxygen, it's rapidly decomposed. So we assess the amount of aggregates. This is just, in this way, it's just all particles which are larger than 2 millimeters. And then when you sieve the soil, it's a wet sieving procedure. You see that this amount declines rapidly, also after 20 years, and then there is new steady state equilibrium at a low level. So the structure is deteriorating within 20 years, and this explains, obviously, the loss of the carbon in the first 20 years of cropping. Now the question is, why do we have this loss of carbon if the yield is stable and if also aggregation and soil structure is stable? When you then go back to the fields and just look what the fields look like, then you see that a lot of material is blown to the sides. So we had the idea maybe the loss of the carbon is associated to wind erosion. And we know that the wind blows away silt particles, so very, very fine dust particles. Then we see that this, indeed, the silt-associated carbon is lost linearly with time, which could be an evidence of wind erosion. Or it's an artifact, because the farmers may have decided they take the soils under cropping first, which have the least silt content. Because in Africa, it's different to here. A sandy soil, you have more rapid infiltration of water. And when it gets very hot and dry, the evaporation from the sandy soil is not so intensive because the water is already at a deeper depth. And then this water is stored on a dense iron concrete layer. So in the summer, the plants can use the subsoil water for growth. And that's why in the South African systems, the sandy soils with less silts are the more fertile ones, just opposite to here, where you like the silt to have the nutrients attached. So in order to decide what is now the process, we looked for an element which is not occurring in soil, and this is the plutonium from the bomb test. And if you look for plutonium isotopes, you can even identify the source of the bombs. These isotopic values didn't change, so we could group them together. All bombs have the same signal. And since we can assume that with the fallout, everything is falling at similar intensity to all fields, we can use the loss of the plutonium as an evidence for loss of soil by erosion. And then we see we get the same loss curve as the carbon, which is a clear evidence that the main mechanism of carbon loss is possibly not even the degradation of the carbon, but mainly erosion. At least in the long-term run, it explains by 100% all processes observed. The interesting question we then had is, can this be reverted? So if you put back the soil under pasture management, you stop plowing, is the carbon coming back? And because of market prices, farmers started to do so 40, 50 years ago. So we get sites of different ages of secondary pasture fields where they reseeded the natural grass mixtures. And then we could look at the soil properties, how do they change with time? And then we find, for instance, that the soil structure, the aggregates, they come back after 10 years. and then the soil structure is restored. So the function of soil to store aggregate inside three-dimensional units is restored within 10 years. And then we thought, okay, then also the carbon should come back. So structure regenerates, and then we looked at the carbon. This is the carbon stock only in 5 centimeters. We see it comes back at a slower rate, takes maybe 20 years. In some ecosystems it's a bit faster, so it's very ecosystem specific now. And it's not completely. So the reformation of the organic matter or the humus in the soil is incomplete. It's only restricted to the surface soil, and the subsoil do not respond at all to land use change. So once this resource is degraded, it is lost at least for more than 100 years if the subsoil is degraded. And thereafter we developed now other techniques to understand the contribution of the subsoil to plant nutrition. We do it, for instance, with phosphate isotopes, because whenever a microbe takes up a phosphate molecule, the phosphate is toxic to the microbes. So it stores the phosphate as polyphosphate and exchanges the oxygen with the oxygen of the cell water. And then the isotopy of the phosphate changes. And you can calculate an equilibrium value, which is the value which is typically occurring in microbes. And if this value is in excess, every phosphate has been seen by microbes. So this is phosphate which is bioaccessible and which is heavily recycled. This is Klein-Altendorf soil. And in the subsoil at a certain depth, we are far away from this equilibrium, so the phosphate is not seen. It's not recycled by the plants. It has a long residence time, and in between there's some kind of transition where we can detect the impact of the root on the acquisition of subsoil resources, which goes down to 60 centimeters. Below that, the subsoil doesn't really contribute much to plant nutrition. And then we tried within the Soil 3 project to manipulate the access to the subsoil. The idea is we make small furrows. We remove the surface soil in order not to dilute this surface soil with other matter. Then we inject organic compost into the subsoil, mix it with the subsoil in stripes, and then put back the former A-horizon, which is not affected by the subsoil loosening. The idea is that we have subsoil with a low density mixed with organic matter so that this effect is long-lasting, and to facilitate the roots to grow into the soil and to take up the water when we get a dry summer. And the technique, which is done by Schmidtmann, works. We see that we get down to 56 centimeters, we get an incorporation of the compost into the subsoil. And when we monitor yields, we see that indeed over these stripes we get a response of whatever plant, wheat plant or barley plants, whatever we plant here. I think this is barley. And when we calculate this on a hectare basis, we get a 20% yield gain in Klein-Altendorf at these very good soils. We did also gain 20% in the very dry summer plus, and now in the third consecutive year, these plots have a 20% yield gain. I think it's something worth to be tested in Strukturwandelgebiet. It's also worth to be tested in Klein-Altendorf. The important thing why I want to mention it, it's a running long-term experiment. It's a 9-year project, so many people are working there. We are using labeled fertilizer studies, and also to understand, for instance, what's the fate of nitrogen, where does it go to, how does root architecture develop, how is water uptake, what is nutrient efficiency, etc. So it's running, will still run for more than 5 to 6 years, and we would be pleased if some PhenoRob sensors would also like to have a look at the field where we know all the heterogeneity of the subsoil, where we monitor already plant growth, etc. So there could be some synergies.
Is this plugging done once or every?
No, we want to do it only once. We know that if you loosen the soil in the subsoil, you can increase yields, but we also know that this effect is gone after 2 to 3 years. So actually, the energy cost to do so does not count. You invest more than you gain back by the higher yield. So what we wanted to do is we wanted to loosen it and then to prevent that the soil can close it again in order to have a long-lasting effect for many years. And that's why we have to monitor it for 10 years. But 3 years we're now successful, so it's quite promising. Then I come to the final aspect. I think this is the advantage to have the discussion in between. How to assess the heterogeneity in the field, because it can be very heterogeneous. Sometimes we have clayey sites, sometimes we have loamy sites, sometimes we have sandy subsites. So the easiest way is we sample very many different spots, put them on these plates, and record a mid-infrared spectra. And then you convert these spectra by statistical methods into, for instance, main components which drive the multiplication of the signal or the diversity of the signal. The different procedures around, like partial least square analysis, neural networks, support vector machines in order to use the whole spectrum information as a predictor of the soil property. And since the spectral assignment is very fast, it's done in 5 minutes, if we would have a model to predict the soil properties, we would have completed the soil analysis very cheap, because that costs only electricity within 5 minutes. And I show you only the data for carbon now. We see that these multiplication factor here, the principal components, they vary between different zones. We did it for many soils of the world. So an overall prediction for the whole soils and all soils of the world is difficult. We better have to use a regional or a local calibration. And if we do so, that's very accurate. We see here this is an R-square of 99%. And actually, the assessment of carbon is as accurate as the much more expensive chemical analysis. And we could do it for different soil properties. We can do it for pH, for clay content, for oxide content, even for some phosphorus forms. And then we have a very fast mapping of the soil heterogeneity. The disadvantage is it's only valid for one site. We still do not really get the clue to get a global calibration data set. We now have access with students to a database of 16,000 analyses, but still the global model is not yet satisfyingly running.
This is infrared, you said?
Yeah, infrared, mid-infrared. Mid-infrared has a bit longer range. Usually people use near-infrared only. We use mid-infrared in the mid-infrared range.
Do you get the same correlation then for other nutrients besides carbon?
For carbon it works well. Phosphorus is difficult because phosphorus has no infrared response, so this is a spurious correlation. We get it good for nitrogen. We get it for mineralogy and cation exchange capacity, so quite for a range of soil properties. So you could map a rhizosphere, for instance, and derive these properties.
But nitrogen has no spectral absorption.
But 95% of the nitrogen is organic nitrogen.
That's right.
And that's why. And I think that's also why it works for phosphorus. But anyhow, you can also do it within a sensor with reflection light. Stefan built a chamber where this sensor is inside this dark chamber so that it's not interfered by sunlight. That was done in the crop sense. And then you get different spectra. They're not so well resolved, but you also see they vary with soil moisture, so it's much more difficult in the field. But if you do a pre-classification, you use the same spectra also to predict moisture, and then you use a moisture-specific local calibration, you can get access to the nutrient patterns, for instance. The problem with these infrared techniques is it's only the very surface soil. It's only 1 millimeter you detect, and you cannot look inside. So the other technique we are using is gamma spectroscopy, gamma spectrometry, where you directly count the radioactive decay of certain nuclei which are in the environment at a natural abundance, where you can detect this radiation. For instance, of 40 potassium. And then you get a direct tracing, for instance, of the potassium content in the soil. And the intensity of the sensor is about 30 centimeters, so you map immediately the plow layer. Such a device can be mounted on the tractor. And then we see, for instance, that if you do it stop and go, you go, stop, measure, go, stop, measure. You can predict quite nicely already the clay content of the soil. If you take that this analysis of clay is quite time consuming, the whole procedure takes maybe 2 weeks. You do maybe then 30 samples in one run, but this is quite convenient to have it stop and go. If you do it on the go, you have some bias where there's now work done sorry, to solve this bias. You can even do it from the air. So you fly, for instance, with a helicopter. I do not know whether it works for the drones. I think, how heavy is it? 7 kilo? More. A bit more than 7 kilograms. So it's a bit heavy for a drone. But if you fly over the field, you get these little spots. You even could use it when their plants are growing because the signal is going through the plants, and then you would have an in situ immediately mapping of the whole field. If you fly too high, then the spot size is too large and the resolution may be too low, so you cannot resolve the structures. So the lower you are on the ground, the smaller is the spot size of the measurement and the more accurate the resolution. But anyhow, we did it with BGR. And then you see, for instance, in the Transregio, how you get a map like a transect, the flight line of the helicopter, to measure changes in potassium content or clay content across the whole region. Current techniques are now designing to solve these interference of these different spots of measurement. The procedure which is used here is a moving window smoothing. So you don't use one point for calibration, but you always average 5 points. You average 5 points here, then you use next 5 points here, and thereafter you try to get rid of the noise. And if you do so, you get some quite nice predictions of the clay contents. This is one field site in Ascheberg. And if you do it on another site with other mineralogy, you see the global prediction doesn't work. We think that we need a second sensor to detect mineralogy, like an element sensor, which we put here on the invest list. You see, so if you can put the different things together, a simple prediction across different sites is not very feasible. It improves with the use of support vector machines because they seem to be able to decide at which site in advance which calibration is to be taken, but still there are some outliers, and I think this is just future work to improve this prediction within one model across different sites. This is why the soil science part is in CP5. It's still not universal, and maybe it can be improved by other sensors. And with this I come to the end. We think a soil science contribution to PhenoRob could be with the use of endoscopes to understand the patterns of soil root contact zones. Is that different if the root is in contact with soil, or why does it avoid other parts for growth? I would be personally interested in a mapping of soil structure. I don't know whether this would work with a laser scanning. We didn't put this into the proposal so far. And we would like to extend the soil maps to depth, maybe using borehole gamma spectrometry, which then has to go to the supercomputer. So this is why the soil science is not taking lead of such projects, but it can be done in collaboration with Jülich in order to get 3 or even 4-dimensional soil maps. And with this, I thank you for attention.
Yeah, very nice. I was actually going to ask about mapping soil structure. I wondered if do farmers, like including in Africa, for example, do they actually have a way of looking at aggregate stability in soil structure as a part of their
I think they don't do it.
Right.
They can't even protect it because they don't have enough straw residues, so it just decays and flies away.
So how, when you say mapping soil structure as a future technology possibly done invasively, what signal would you use for that? How would that be done?
Well, the perfect way would be the field CT, whenever this works in a near future or never. I just put it into the proposal because you said you were working on it. A computer tomograph working in the field. That is the far-off vision in 30 years or 50 or 100. And I think we should see the structure by either resistance when you go through it, a surface like a lateral penetration resistance, and by surface roughness. We have to translate the roughness information at very high resolution into soil structure information. So I think if we would have very high resolved laser scanning on the ground, we should be able to monitor structural dynamics by a sensor.
What means very high resolution?
At best, at least a tenth of millimeter, 100 micrometer. But that's just guessing. Yeah, Gabriel.
I still don't get my head around that there's so much carbon loss by wind erosion. It's kind of mind-boggling, I find. I mean, you use plutonium as a proxy for wind erosion. I'm just wondering, is there no plutonium uptake in plants? And actually part of the loss of the plutonium is actually just because of harvest takeaway from the field, basically? Do you measure plutonium in plants?
No, we did not. But usually plutonium is thought to be very immobile, same as cesium. But there's literature around.
But you find cesium in this upper part of certain mushrooms and stuff like this, right? And there's uptake into organisms with these metals, right? And I mean, I don't know what plutonium.
Yeah, but as far as I know, the literature, there may be uptake in the roots, which doesn't matter, or even absorption to the roots because they remain in the soil, but that it is really transported in significant amount into the top plant organs which you harvest. We should do the exact calculations, but as far as I know, this is not a relevant process which can really explain these losses.
Two questions of this carbon balance. You told us that it takes the first 20 years of agriculture, then you lose more or less the half of the carbon. So I think this depends also from the kind of agriculture. How do you do it? Or is it
exactly. Yeah, exactly.
What is the data? And the second question is, what you are thinking about the agricultural soil changing as a sink for carbon? For example, if you increase the organic carbon into the agricultural soil in Germany for 1%, estimation says more or less 950 million tons CO2. This is a little bit more than what we bring every year into the atmosphere as the German. What do you think about this? Maybe some estimations will take at least 5 years. So what do you think about carbon sequestration as a climate effect and what we can do there?
Yeah, I must confess I'm quite skeptical. Next week is the climate conference, the 4000 conference in France. Will also be there. You can increase carbon at best by organic fertilization with more or less stable organic fertilizers. You can do it a bit by intercropping. You can do it by reduced management to a certain degree. So you can gain at certain ecosystems where you have these fertilizers available. You don't have in Africa, but you may have it here. You can increase the amount of carbon which you want, but it means you increase the N ratio. You increase the stress of oxygen being used up in small microsites. So I think it will affect negatively the nitrogen balance. You get the winter mineralization, which is bad for the nitrate problem, and you get more N2O into air. And there's still a controversial discussion whether the carbon amount, which is, for instance, sequestered in the soil by zero tillage, is not offset by increased release of carbon equivalents as N2O, which I think often happens. Yeah. So I think you get the carbon in, but you get N2O out. And since one molecule of N2O counts like 320 molecules CO2, 2 kilogram N2O is sufficient to compensate for 600 kilogram carbon sequestration.
But the converse tricky thing is that there's been a lot of focus on also biopores and roots following previous, whether they do or don't follow previous channels. And in general, when they follow the channels, actually the leaf growth and plant performance can be lower.
We agree.
People look a lot at that, and one of the big reasons seems to be, it could be allelopathic effects, but it also could be contact. So the core size and interaction in space.
Yeah, it could also be, it could also just carbon loss. If the plant invests too much carbon in roots, it's also lost. But we think that these biopores, they have an abundance of maybe 5%. So they are only affecting parts of the roots. So and it is not to increase the yield, the biopore, but it's increase resilience against drought. Because once the root is deep in the soil, if it is dry, it can take up the subsoil water, and then you get the yield gain. If it is not dry, you have invested it wrongly. You don't need it. So it's more like we don't think the subsoil should be used to increase yield, but to prevent yield loss at hot summers. And it's more like building up an insurance system. And we always have this discussion, which genotype do you need? And likely you need a genotype which does not just deeply grow, but which has 1 to 2 or 3 roots going deep, and the rest stays in the subsoil. But just to have, at best, just roots down there without much soil-wood contact, in case they need it, they can switch the nutrition strategy. That's from our perspective, the idea. Yeah, just in time. Thanks.